Bismillahirrahmanirrahim. In this video, I'm going to discuss the neural mechanisms which enable our brain to control and express pain sensations. Let's start. The degree to which different people react to pain varies tremendously. This variation results partly from a capability of the brain itself to express input of pain signals to the nervous system. The brain does it so by activating a pain control system called the analgesia system. The analgesia system consists of three major components: the periaqueductal gray and periventricular areas of the mesencephalon and upper pons surround the aqueduct of Sylvius and portions of the third and fourth ventricles. Neurons from these areas send signals to the raphe nucleus. It is a thin midline nucleus located in the lower pons and upper medulla, and the nucleus reticularis paragigantocellularis located laterally in the medulla. From these nuclei, second order neurons are transmitted down the dorsolateral columns in the spinal cord to a pain inhibitory complex located in the dorsal horns of the spinal cord. At this point, the analgesia signals can block the pain before it is relayed to the brain. Several neurotransmitter substances are involved in the analgesia system. The nerve fibers derived from the periventricular nuclei and from the periaqueductal gray area secrete enkephalin at their endings in the raphe nucleus. Fibers originating in the raphe nucleus send signals to the dorsal horns of the spinal cord to secrete serotonin at their endings. The serotonin causes neurons in the dorsal horn to secrete enkephalin as well. The enkephalin is believed to cause both presynaptic and postsynaptic inhibition of incoming type C and type A delta pain fibers where they synapse in the dorsal horns. Thus, the analgesia system can block pain signals at the initial entry point to the spinal cord. The analgesia system is based on the occurrence of several naturally secreted substances in the brain which act as neurotransmitters in various parts of the nervous system. About a dozen such substances have been found at different points of the nervous system. The most important of these are beta endorphin, metenkephalin, leuenkephalin, and dynorphin. All are breakdown products of three large protein molecules, namely pro-opiomelanocortin, pro-enkephalin, and pro-dynorphin. The opiate system of the brain was discovered when researchers discovered that injection of minute quantities of morphine either into the periventricular nucleus around the third ventricle or into the periaqueductal gray area of the brainstem causes an extreme degree of analgesia or pain suppression. It was assumed this effect was produced by activation of receptors, which must be the receptors for some morphine-like neurotransmitter that is naturally secreted in the brain. Therefore, an extensive search was undertaken for the natural opiate of the brain. In subsequent studies, naturally secreted morphine-like agents, mainly the opiates, were found to act at many other points in the analgesia system. Inactivation of pain pathways by morphine-like drugs can almost totally suppress many pain signals entering through the peripheral nerves. Opiate-like drugs are used to treat moderate, severe, and chronic pain. They are also used for pain relief after major surgeries, treatment of cancer-related pain, and shortness of breath at the end of a patient's life. However, morphine and related opiate drugs also have a high potential for abuse because of its pleasurable effects and relative accessibility. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.